Hello, everybody. Welcome. We are here to hear more from UC San Francisco. We have invited Dr. Corey Harwell here to join us and tell us a little bit more about both the research in his lab, as well as the graduate programs, and even a bit about the undergraduate summer programs that are available. And so before I begin, I wanted to tell you a little bit more about our host or our guest. Corey Harwell is the Associate Professor in Residence within the Neurology Department. And he is also a professor within the Eli and Edith Bro Center of Regeneration Medicine and Stem Cell Research. And so with that, I will hand it off to Corey. Please get started anytime.
All right, thank you, Sophia. Well, I'm happy to at least be able to visit you virtually, and hopefully maybe sometime in the future, I'll get to meet a few of you in person. So as Sophia said, I'm currently an associate professor in the Department of Neurology here at UCSF. Been here for about a year and a half. So prior to coming back to UCSF for me, so I was a postdoc here years ago. I was an assistant professor in the Department of Neurobiology at Harvard Med School for 7, almost 8 years. So it's sort of a recent homecoming for me to come back to California and back to the Bay Area. So I thought I would tell you a little bit about my lab and about some of the research that we do for discussing a lot of the programs and opportunities that are available at UCSF. So this is just a picture of my lab, which is currently composed of 4 postdocs and a student and a technician and a lab manager. And of those, about 4 of the people that are currently in the lab are people that moved with me from Boston about a year or so ago. So the major focus or interest for my own lab is understanding this kind of fundamental question in neuroscience of how is it that you get the massive diversity of cell types within the nervous system. And I would argue that the nervous system is one of the most interesting places to kind of ask this question. You can probe and study the development of cell types across a number of organ systems, but the nervous system, in my opinion, is unique in that the diversity of the morphologies, the functions, the connectivities of these different cell types is essential for, it's a fundamental feature of the nervous system for its complex functions. And I'm just showing you here a recent part of the brain that we've become especially interested in studying. And you can probably appreciate, if you can hopefully see my pointer in this drawing, the diversity of cell types in the structure that sits in this part of the medial part of what's called the forebrain. And this is just a section of an adult mouse brain. And outlined are these 2 structures called the lateral septal nucleus and the medial septal nucleus, and MS. And then in this cartoon, you can see examples of some of the diverse morphologies of the cell types and the neuronal cell types that are found within the lateral septum. And there's actually a number of other reasons that we've become interested in the septum. So in addition to its diverse morphologies, it also has these complex patterns of connectivity, and it's really highly interconnected with regions of the brain that are involved in, say, higher cognitive processing, such as a region called the hippocampus and prefrontal cortex, which you may have heard about for any of you that have neuroscience interests. But it's also highly interconnected with these limbic structures that are involved in regulating innate behaviors. So it's thought that the septal nuclei function sort of as a node for integrating kind of higher cognitive information, and emotional and affective information, and kind of incorporating these two streams of information to facilitate appropriate contextual behavioral responses. So, given that idea about its function, it's probably not surprising that there's evidence that dysfunctions in cells within the septal nuclei are implicated in a number of neuropsychiatric disorders, including psychotic spectrum disorders, depression, bipolar disorder, schizophrenia, etc. But our interest is in that basic fundamental question of how is it that you get all of these diverse cell types and complex patterns of connectivity between the neurons within the structure. And some of the ways that we go about classifying the cell types is by we use mouse models as our model system, is that we classify cells based on where they came from. So during embryonic development, all of these mature cell types are derived from different progenitors and have specific regions in which they are born. Their molecular identity, so we can use a number of tools, including, this is an example of a plot using a technique called single cell or single nuclei sequencing, in which we can profile the gene expression patterns of individual cells, and then use these different algorithms for actually plotting or clustering cell types that share common molecular identities. Oops. And then also just looking in the structure at the anatomical distribution of these diverse cell types tells us something about, the cells they interact with and their particular position or role within the neural circuit. And we can also use tools to genetically label these cells to start to interrogate their patterns of connectivity. So another aspect of their identity is how they're connected to other cell types, and for these cells in the septum in particular, they project to structures outside the septum to make the majority of their connections. So that's another kind of dimension of their identity that we use to classify cell types. And then I've already touched on some of this from the previous drawing, but their morphologies also tell us something about how the cells function and other cell types in which they might interact with. And then because these are neurons, the other important feature is since these are electrically active cells, they have distinctive patterns of firing for each of these distinctive groups of neurons. That tells us about the specific patterns of communication between these distinctive neuronal cell types. But so we've done a lot of work since the septum was such an understudied structure and just kind of classifying these cell types, but ultimately our main interest is how is it that these different cell types are actually specified during development. And again, we rely heavily on the power of mouse genetics, which allows us to use different mouse reporter lines to genetically label these different groups of progenitors. Typically, these progenitors are defined based on the expression of distinctive transcription factors. So for example, we can classify different septal progenitor zones based on either the exclusive or combinatorial expression of just two transcription factors here, ZIC4 and NKX2.1, which are just two different families of transcription factors that are expressed in these septal progenitors. And I'll show you what that looks like in the embryo. But so this is what we call genetic fate mapping, where we can label those cells that express either ZIC, NKX2.1, or NKX2.1 and ZIC here labeled in green. And trace the fates of those cells over developmental time to see what they will ultimately go on to become. And we also use approaches such as, again, single cell sequencing, where we can also look at the molecular programs that are involved in the specification of those cell types by collecting the embryonic and early postnatal septum over developmental time, and then transcriptionally profiling the changes in gene expression of those cells. And that allows us to produce these maps, or we call inferred trajectories of the specification of these distinctive neural cell types. So we start to at least get a picture of the molecular programs that go into the specification or maturation of that cell to its final mature type. And recently, we've also become really interested in not just the specification or diversity of neuronal cells, but also non-neuronal cells, and in particular astrocytes. And we've learned a lot of really interesting things about astrocyte diversity in the course of our studies of the septum that were somewhat unexpected. And astrocytes, traditionally had been thought to be these kind of passive support cells that were the glue of the nervous system that just kind of produced the scaffolding in which the neurons use to connect to each other. But in the past, 15 or more years, the view on the role of astrocytes in particular and their roles in both helping to sculpt the formation of neural circuits and also modulating the activity of neural circuits has really grown. So we know that astrocytes have prominent and important roles in nervous system function, and still supporting neurons with ion homeostasis, uptake of neurotransmitters, and during development, they actually have prominent roles in regulating synapse formation and synaptic pruning. And as I mentioned, there's been a relatively recent appreciation that astrocytes are nearly as diverse in their functional and molecular types as the neurons in which they support and coordinate in these brain circuits. So, some of the fundamental questions that we've become interested in is, how is it that these astrocytes in particular are specified? And we've been looking at this. I was talking to a student yesterday, and they brought this up, the idea of kind of the nature versus nurture, or how we look at it as, what are the intrinsic mechanisms? So what is it about the developmental origin, the progenitors that a particular group of astrocytes or cells come from, that goes into programming their identity, versus the extrinsic factors, which are the environmental cues that will actually shape their specification. And this is turned out to be a really interesting question to pose within the septum, because we've identified at least two major molecular types of astrocytes that also have distinctive developmental origins from those different NKX or ZIC expressing progenitors. And yeah, so here's an example of that differentiation of where these cells are coming from that express these different transcription factors. So here's just a section of an embryonic mouse brain expressing a fluorescent reporter for ZIC4 Cre, outlined in green, and labeled with an antibody actually against NKX 2.1. And so the septal, the rostroseptal proliferative zone is labeled in green. And that's where a majority of those lateral septal astrocytes and lateral septal neurons are actually derived. And then there's a region in more caudal parts of the septum that co-expresses both ZIC and NKX 2.1. And this is a region that we start to refer to as the septal eminence, that produces a very distinctive subset of lateral septal neurons. And then as you go even more caudal, the names of these regions is important, but there's a region that exclusively expresses NKX 2.1 that's involved in producing most of those medial septal neurons and astrocytes. So based on a lot of our genetic tools and our fate mapping, we've been able to kind of start to build a map of the fates of these distinctive cells so that we know that NKX 2.1 cells produce those medial septal neurons and astrocytes. The cells that co-express both NKX 2.1 and ZIC produce lateral septal neurons. And then the majority of the other lateral septal neurons and pretty much all of the lateral septal astrocytes are derived from those ZIC-expressing progenitors. And we've begun to explore in the same way with the neurons, with astrocytes, the different dimensions of their differences in these cell types. So there's already distinctive morphologies of those medial septal and lateral septal astrocytes, where the medial septal astrocytes have somewhat longer processes, longer branches, and are somewhat less ramified or bushy compared to the lateral septal astrocytes. And then again, we wanted to look at the molecular diversity, since we know that there's this distinction between the contribution of NKX 2.1 to the medial septal types versus ZIC to the lateral septal types. We can actually combine our genetic fate mapping by, again, using our Cree reporter, where we can sort those cells that have a history of NKX 2.1 expression from those cells in the septum that come from the ZIC expressing population. And this allows us to kind of combine our fate mapping with our single cell sequencing so that we can build these maps of the gene expression profiles of the different cell types along with their developmental origins. And here we can resolve what we've seen with the anatomical fate mapping, which is that there's also distinctive molecular differences between astrocytes that are derived from NKX 2.1 progenitors versus the NKX negative cells. And if we look at genes that are enriched in those distinctive groups, we can find that there's these really beautiful segregations of the genes that are enriched, say, in this example from the NKX 2.1 progenitors that specifically outlines medial septal astrocytes. And we can do the converse for the ZIC-derived astrocytes. And again, there's different molecular markers. This time it's for glutamate transporter, GLT1, which is highly enriched in those lateral septal astrocytes. And we don't see nearly as high of enrichment in the medial septal. But part of what we're really interested in is how much of these molecular differences are based on the progenitors that those cells are derived from, or where they come from developmentally, versus the cues that they encounter in their distinctive regions. It's because we know that medial septal astrocytes interact with a different class and a different group of neurons than the astrocytes within the lateral septum. So what is it that might be coming from those distinctive groups of neurons that could also be directing the molecular specification of those cell types? So this actually kind of touches again upon our fundamental question of the intrinsic factors versus those extrinsic neuron-derived cues. And the way that we've begun to start to answer that question is by, again, utilizing our sequencing, but this time looking over developmental time during the specification of those distinctive groups of astrocytes into their mature cell types. And this is, we've produced this pretty big data set of, it's got now 166,000 nuclei. And again, we've used our NKX 2.1 Cree, where we can overlay both our fate map with our transcriptomic profiling. So we can see how the developmental origin already specifies these molecular differences between these cell types. But in collecting these cells from the septum over developmental time actually allows us to kind of look across two dimensions, so that we can look at the genes that are differentially expressed between NKX-derived or positive astrocytes in the medial septum versus the negative astrocytes in the lateral septum. But we can also look at the differential enrichment of genes that are expressed in these astrocytes over the course of their maturation. So starting at P0, when we know that astrocytes aren't fully specified yet, we can start to look at the genes that may be involved in either getting them to the right place, or the factors that promote their interaction with other cell types, that are neighboring them within each of these structures, that may be important for their ultimate mature cell type identities. So that's sort of, I guess, maybe a little vignette about what my lab is interested in, and just kind of what we're thinking about in future directions. One of the exciting things about studying the septum for us is that because it's involved in regulating these innate behaviors related to anxiety or aggression, and that we've begun to classify these different cell types, both the neuronal and the glial cell types, we can start to address how particular cell types might be responsible for mediating very specific types of behaviors. And then also using a lot of our sequencing, which we use as a tool for hypothesis generation, we can start to ask, what are some of the molecules or factors that are involved in wiring up, producing the very precise circuits that are necessary for the function of circuits in the septum. And then finally, we're interested in not just the genetic, but also the epigenetic regulation of these complex gene expression programs that are important for specifying the identity of all of these cell types. So I'll stop there with the science portion, just in case anyone has any questions.
Yeah, so if there's anybody in the audience who has questions for Dr. Harwell, you can either raise your hand, unmute, or feel free to type it into the chat. But we would love to hear if you have any questions about Dr. Harwell's work. And in the meantime, at least I wanted to say hello. I didn't actually get to introduce myself. My name is Dr. Sophia Taineri, and I am working in the Undergraduate Research Hub as the Research Scholarships Coordinator. So we'll tell you a little bit more about our office possibly at the end, but just wanted to give you a minute in case someone had questions. And if there aren't any questions, we can certainly move on to the portion to talk more about UCSF Graduate School and the summer research programs they have available.
Oh, and also feel free to put questions into the chat as well.
All right, thanks.
So can you see these slides? Great. So so actually gonna switch gears a bit and then talk about some of the opportunities at UCSF, both for undergraduate and postgraduate opportunities for UCSF, and also the graduate programs, which I hope many of you are going to be interested in. And I can talk a little bit about some of the programs from direct experience. So I was telling Sophia that I was actually a summer student in this research training program, and in this exact program in the SRTP. Quite a number of years ago now, but I think that the program has probably only gotten better, and I had a chance to interact with some of the recent SRTP students this past year, and I can speak myself actually for the benefits of this program, particularly if you have interests in science and pursuing graduate education in basic sciences. So yeah, so the summer program is a comprehensive program, and it does actually provide you an opportunity for sort of a preview of what life will be like in graduate school. So what it's like to do research full time, at least for a period in the summer. And typically you're paired with a graduate mentor. It's oftentimes you're working directly with someone in the lab who's either a postdoctoral trainee or even a graduate student over the course of the summer. And then there's other opportunities where you'll get to actually interact with other components of the community, where they'll have workshops. And actually, this was my interaction this past summer, where I was involved in organizing these neuroscience-focused lunch talks for the SRTP students this past summer. And then from that research, at the end of the program, you'll give an oral presentation, and we'll also put together a poster presentation. And actually, I can attest to the value of having the opportunity to both do the research and to get the support to put together the presentation for the research. So it was actually from my UCSF summer experience poster presentation that I got the opportunity to participate in another summer program that eventually paired me with my PhD advisor. And then I think another advantage, particularly for those that are going to be interested in graduate training, is to forge those relationships and to have the recommendations that will be a part of your graduate applications from a faculty who's had a chance to interact with you and get to know you over the course of the summer. Yeah, so I think I talked about that. Oops. Yeah, I think there's also kind of a fun networking aspect to this, where you'll get to know a group of students that share common interests and common ambitions from all across the country, and you will live together, not together on the same place, but usually you're housed. I think for the summer research program, you're housed at USF, which is just up the street, and their dorms for the summer. And again, I think that this can't be understated as the opportunity to kind of forge those relationships that will form networks that will help you put together what will be a even more competitive graduate application. I can definitely attest that the graduate student advisors are great. So again, a lot of my interactions with the program was in partnership with one of the graduate student advisors and helping to organize these lunchtime talks. And I think that UCSF, for me, and one of the reasons that I was excited to come back, is that I feel like there's a critical mass of pretty young faculty. No offense to the older faculty, although I'm probably somewhere in between now. But that, with those newer faculty, I think there is like a level of enthusiasm and investment in young scientists that's crucial, I think, for a lot of you at this particular stage in your development as a scientist. Okay, eligibility. So I think and people are probably aware, the major thing is that these programs aren't geared for students that are interested exclusively in medical school. They're typically geared towards students that are focused on getting a PhD or possibly an MD PhD. And then eligibility for this particular program is for US citizens or permanent residents. And it's typically for these programs that I think they're most beneficial is between your junior and senior years. So that would be the ideal time to apply. And what is the application made of or composed of is you'll write a statement of purpose, talking a bit about your interests. I think that it's completely appropriate and encouraged to reach out to faculty and express your interest, and maybe even set up a time to have an opportunity to learn more about their research. And then you'll need letters of recommendation from either if you're doing research on campus currently, from your faculty supervisor there, or from faculty that could give, I think, a strong letter that's relevant to the components of to the focus of the program. So typically, it's going to be focused on biomedical sciences, then maybe one of your professors in which who's gotten to know you related to your interests in biology or chemistry or physics or even computer science. And this is important and something I have to remind not just my students, but the postdocs in my lab, which is that you want to ask people for recommendations with enough advance time so that they don't feel like they're getting things at the last minute. Because I think oftentimes people want to write, if they're willing to write you a letter of recommendation, they want to write you the best letter that they can, and you can help them do that by giving them enough advance notice for putting those things together. And then I think this is a little bit either virtual or in person, depending on COVID. I'm not sure if that's, I don't think that they do an interview that I know of. So I think maybe they're talking about the actual program, and this is probably dated for other COVID times. But as far as I know, all of the components of the program are in person now. And please do check the websites for even a more explicit information. So the deadline for the SRTP is coming up. So it's November 1st. Oh, excuse me, the application opens November 1st, and then the final deadline is in February of 2023. I'm thinking of graduate applications. And the program will run between May 27th through August 4th. And they're aware that students that are on quarter systems may have to start a bit later, depending on how their classes and exams end. So select about 40 or 50 students, which is much larger than the SRTP program when I was doing it almost 25 years ago. And they, I think, typically solicit volunteers across the campus for evaluating the applications, and that they take a holistic approach, which is similar to the approach also taken for evaluating graduate applications. So, all of the typical things that went into the application are considered, but it's not that one thing will guarantee or, preclude your application from being considered. And then here, I hear this used at UCSF quite often, where they talk about the distance traveled, where it's also taking into account the circumstances and addressing this question of, with the resources that were around you, how is it that you were able to excel? Or another way that I've heard it put is like your trajectory, your development, as you progress through your education and through your scientific training. Is it's not expected that you needed to start off to be, say, a straight A student from the beginning, but are you on a trajectory where the evaluators can see that you're going to kind of continue that steady growth? And so they take at least one student from the schools that they recruit from. I think that's no surprise that almost all of the schools in California we hope to have represented, but there will also be applicants from institutions from all over the country. And they do, I think that the program does most of the matching of students with supportive faculty, and I think both their research interests and their prior research, and even potentially the particular faculty themselves and their availability are all taken into account. And I have a bit of a funny story about this, which is the reason that I'm a neuroscientist is because I was somewhat, as far as I can tell, from my view, randomly assigned to a neuroscience lab during my summer research program at UCSF. And I came to UCSF, I was a chemistry major in college, so I had no neuroscience background, but someone with, I think, better perspective than I at the time put me into this lab that worked on the neurobiology of C. elegans, on worm olfaction, actually. And it was that experience that actually sparked my interest in neuroscience that I've carried from, and actually provided me that exposure. Without that opportunity, I actually probably would not have even known or even thought to pursue a career in neuroscience without that experience. Yeah, so you'll be placed in a basic science lab. You'll definitely be supported. I think that this is again a fantastic program that's been running for, I don't know how long. It's got to be longer than I've been around, so 30, 40 years or something. I have to ask Carol Gross about that. But they take a lot of pride in the program, and I think that it's a great place. San Francisco is also just kind of a fun place to spend the summer. And of course, you'll be paid. The pay ranges between $5,000 and $6,000 for the summer. Your travel will be supported, to and from San Francisco. I mentioned that you'll be in kind of dorm-style housing, so you'll be living with the other program participants at San Francisco State, which is actually in a kind of a nice part of the city that's somewhat in between the two campuses at UCSF. So that is the section on the SRTP. I don't know if anyone had questions in the chat. Oops, related to. Yes, so what is the role that previous research experience will play into the application? Do they prefer admissions? Do they prefer applications with no experience? I would say probably not. I don't think that would, I can't think of an instance where having prior research experience and applying for a summer research experience that I assume is going to be external to your current institution is going to be a disadvantage. And in fact, as I read some of the slides. So these slides were developed by the program, is that they mentioned your supervisor, and I think that they were talking about your kind of home institution research supervisor. So I don't think that there's going to be any disadvantage for having some prior basic or laboratory research experience for consideration for the SRTP. Hopefully that answers your question. So next I was going to talk about the postbac program at UCSF. So I think this is a somewhat newer program and is provided for students that may either want or need an opportunity for actually getting more of those research experiences that we're just talking about before applying to graduate school. And the way that they've structured this is in a 2-year paid postbac program, in which, and this is focused in particular on students that are traditionally underrepresented in the sciences. So it's a paid research experience. You're paired with a mentor. It's very focused on the research component of your graduate training. I know some postbac programs have more coursework, and I think that those things are in discussion, but currently it's primarily going to be research intensive. And these deadlines are going to be out of date, but I assume it's probably going to be around the same month. So sometime toward the end of the year is when you'd want to submit an application for the Propel. So this would be for students that are close to graduation, thinking about graduating in the spring, but don't feel like they're quite ready at the moment, or at least want to acquire some additional experience before taking the dive fully into graduate school or into PhD programs. So speaking of the PhD programs, I think UCSF is, you probably know, is a medical school. So there is no undergraduate component at UCSF. It's all postgraduate, which is either the medical school side, so the MD students, or the graduate programs. So the entire institution is focused exclusively on postgraduate education. And I think that that creates a somewhat unique environment for focusing on both the basic and biomedical related research, and also the other aspects of human health directly with the medical students, dental students, nursing students, etc. But I think that it's an outstanding institution. I mentioned about the faculty that, that there's a number of world-class researchers, research institutions that are affiliated with UCSF. And I think that there's student presence across all of those programs. Thank you, Sophia. It's to everyone. Yeah, that the applications for the Propel program, the deadline is January 6th. And there's a number of different programmatic options. So going from bioengineering all the way to oral craniofacial sciences, pharmaceutical sciences, and then some of these, I would say almost all of the programs are interdisciplinary. And I think part of that is just kind of how modern science is conducted. But some of the programs like TETRAD are particularly focused on kind of the interdisciplinary components of these different basic science disciplines. I can speak more so on the developmental stem cell biology program and the neuroscience programs, which I'm more involved in. But I think that for all of the programs, and actually this is for students thinking about applying to programs, is that it isn't a bad idea to consider and evaluate more than one of these programs, because often, just like me, where I'm in the developmental stem cell biology program and the neuroscience program, your interests will also span across the focus of a number of these programs. So it's good to take a look and evaluate the different components and aspects of the programs that might best fit you. And really the differences will be in the first couple years of the program, since once you get into a lab, once you start doing your thesis research, it's relatively similar across nearly all of these programs. But some of the aspects of the training, coursework, and other requirements, there are some subtle differences. Right, so as I mentioned, this is probably the place where those programs are most differentiated in that first year, and parts of the second year where some students are still taking elective courses. But then from years 2 to 5, where you pick the lab, you're basically finding and working on your thesis project, and are still part of the program, and there's other supports from the program, whether it be through retreats where you'll still have an opportunity to interact with other faculty in the program, or through seminar series that are supported by a number of these programs. And then, of course, all PhD students are supported by a stipend. I assume that's up to date for 44,000, but they adjust it yearly, but it says 2021 to 2023. So it may be worth to check back on that. And then, what can you do with your PhD from UCSF? So there's students that are across, all of these different career paths. I would say, one of the places for UCSF students that is maybe a little bit unique compared to, other parts of the country, and even other parts of California, is because of the large presence of biotech around the area, is where students are, I think, have opportunities for exposure and for exploring those career opportunities relatively early on in their training. So the deadline for, I think, all of the PhD programs should be December 1st. The MSTP program, I guess, that has already passed, and the DDS PhD program. And I'm not sure if any graduate program at this point requires it. I guess there might be some holdout somewhere, but UCSF graduate programs do not require the GRE. And I think that for most of the programs, there's not even a mechanism for submitting the GRE, even if you had taken it. And if there's financial need, UCSF has mechanisms in place to waive the application fees, which is, I think, an important consideration thinking about now, where students are applying to 8 or 10 programs, which can quickly become cost prohibitive. And I think the rules for requesting letters of recommendation is similar to, so you can think about the things that for the summer research program are sort of the practice for what you're going to do when you actually begin applying for PhD programs. And this is where that advantage of having participated in summer research programs comes in, where you can have not just your home institution professor for which with whom you've done research, but also someone from outside your institution in which you've spent a significant amount of time in their lab, who can discuss your potential as a scientist, which I think makes for a pretty compelling letter of recommendation. In contrast to having, one from a research supervisor and maybe a few from professors from your institution for which you've taken courses. And same rule applies in the more advanced notice, the greater opportunity you're giving them to write you the best letter that they can. And I think the other components are pretty typical, where you'll submit your transcripts, and again, writing a statement of purpose, also similar to the summer program, but this time more involved. And why is it that you want a PhD? What are some of your accomplishments? And again, with all of these things, I think across nearly all of the programs, UCSF takes a holistic approach with each component of the application being considered. And then the next stage of that, so after you've done your applications, you're invited for interviews. I think this is a pretty typical structure. It'll vary by institutions, but typically you spend at least a day, if not 2 days, visiting the institution, in which you'll have time to interview directly with faculty for some period, somewhere between 20 to 40 minutes, I think is typical. And then this is, I think, more recently incorporated into a lot more programs, where you also formally interview with a graduate student. And then there's typically either a research symposium or research presentations from different faculty in the program to sort of illustrate some of the kind of breadth of research directions within any of the particular PhD programs. Yeah, and I think this is a common structure where you have kind of lunch with students that might be a little more relaxed during the time that you're doing your interviews. And then you'll have dinner with faculty, which should still be relaxed, but I know that it's always kind of a different level of stress when having dinner with the faculty that are also going to be involved in evaluating your candidacy. And hopefully programs are making adjustments in the other way to make things more interactive and more in-person interaction opportunities at this stage in the pandemic. So there's a number of, I think, really great resources at UCSF for supporting diversity, equity, and inclusion. I don't know for all of the graduate programs, but for neuroscience, in which I'm most heavily involved, there is a DEI committee, which I'm also involved, that addresses a lot of issues, and is actually, I think, been pretty well supported by the director of the program in taking a lot of advice, and has actually the students, and actually not just from the DEI program, but I think the students in general have had a lot of impact on making changes to the policies for, how the graduate program interview day is structured. There's now students that are on the admissions committee. So I think that all of those things have helped to both increase the transparency and openness of the process while still, respecting the confidentiality of people's applications. But I think at least to provide the feeling for students that that there's fairness in how the applications are being evaluated, and then just broadly more across the program, how programmatic decisions are being made. But then there's this really fantastic DI leadership course that's provided for graduate students that I think is really heavily subscribed. And also the same for the race and racism and science course, which I think, as far as I know, all of the program will have opportunities. And then there's the support from the programs I talked about, Propel. I'll talk in a moment about IMSD and the ARACTA program for postdocs. And then there's a number of registered campus organizations to support all of the spectrum of diverse groups and interests that you would imagine to need to be supported in a campus that supports both international and national scholars. And I think especially the UCSF first generation support services, that is, I think, a pretty well supported office. And I think that there's a substantial population of first generation students in both the graduate and medical and dental programs that are supported by this office, or that have opportunities for support from this office. And then there's a number of student support services. There's student health counseling. Of course, there's support for LGBTQIA students with particular needs, where I think in San Francisco in particular, there's a number of great resources there. And then I think there's also a pretty extensive mental health services that are available for students, and that there's support and advertisement so that students, as they're coming in, know what services are available to them, which I think is important. It's not just having all of these different services, but making sure that the students know how to access and who to contact to access those services. And then here's just like another example of some of the 150 registered campus organizations, Graduate Career Alliance, SACNAS, and so on. And I should check the time to make sure I'm not running way over. Oh, it's right at 1 o'clock, so I think I'm getting to the end. But these are just other examples of additional supports for students, so support for students with disabilities, and so on. San Francisco is expensive. California is expensive. So, and there's been a number of really great efforts recently to help alleviate some of that, which is that there's a lot of student housing that is typically below market. And it's a mix of for just students and also other trainees, postdocs. So a few postdocs in my own lab live in UCSF housing. have been pretty happy with that. And for students that don't live on campus, there is a need-based cost of living supplement that's provided for students. And then there's additional services where there's food market, there's wellness services that I talked about, and financial aid support. So again, there's a number of resources that are made available to support students with the acknowledgement that San Francisco can be an expensive place to live. And again, please check the websites and find even more specific information. And I'll just talk really briefly about the IMSD program that I've had an opportunity to interact with. And basically, it's a fellowship program for incoming basic science PhD students. And it provides, some financial support, so for instance, funding for conference travel, but also there's a lot of career and professional development and enrichment activities. And also, I think there's a lot of value in the network where, again, this is focused on supporting historically underrepresented students that are part of the basic science programs at UCSF. And this is supported by Deanne Duncan and Jason Sello. And I think that all of the students that I've had a chance to interact with, and actually I've been part of some panels related to IMSD, have found that it helps them to, with the transition and integration into the program, to help some of these students kind of get their bearings. And I, would just go to Deanne Duncan, who's the Assistant Dean of Diversity and Learner Success, and she is a fantastic, supportive resource for students, I think, across many programs, not just for the IMSD program. And if you had any questions about IMSD or any other questions related to diversity, equity, and inclusion at UCSF, Deanne would be a fantastic resource. And Jason is the other co-director of the IMSD program, who's a professor of pharmaceutical chemistry and a great scientist, but also incredibly supportive of the DEI work that's going on across UCSF. And please, reach out, contact any of us with questions, including myself. And I think I got through all the slides. So I'll stop there just in case there are any other questions. I don't think I caught anything else.
I don't see any other questions in the chat, but of course, if you have any, please submit them.
I'm getting them direct. So are there programs available for undergraduates who are interested in medicine and health sciences, though have not yet had any previous experiences in a research lab? Yeah, so I actually have to say that I am sort of ignorant to what are the program opportunities for students that are actually interested in medicine and medical school at UCSF. I think that those exist, and I would have to follow up maybe with Sophia. I don't know if you're aware of any of the kind of more medical health science related programs.
I am not familiar with them, but certainly you can share information and I will share it with the attendees. I see there's also a hand up. Annika?
Oh yeah, I was interested in the graduate programs at UCSF, and I was wondering what the biggest difference you would say between like an institution with UCSF with like no undergraduates and an institution that has an undergraduate population. Like, what's the biggest difference doing research as a graduate student there, or is there not really a big difference?
I think that it probably varies by institution, but I would say, at least my perspective as a faculty, one of the major differences for being in a medical school versus an undergraduate serving institution is the teaching load. So, you know, all of my time that isn't like doing other administrative stuff is really focused on the research in the lab. So not putting together, you know, a curriculum for a course or something like that. So I think that in that way, it is maybe more beneficial for graduate training, since you're going to be in grad school for the research, and that's how I spend the majority of my time as a faculty.
That was helpful. Thank you.
Sure.
Did you have any other questions that were sent to you directly, perhaps? I don't see any in the chat right now.
No, that was it.
Okay. Well, thank you very much. I know we're a few minutes over time, so thank you for everyone that has hung out and stayed with us for a little bit longer. Just as a quick note, here at the Undergraduate Research Hub, we are working to help students just get to know undergraduate research, you know, in any way possible. So we're happy to be a resource for you all. Please feel free to reach out to us if you have any questions. ugresearch.ucsc.edu or ugresearch@ucsd for the email address, .edu. But we're happy to talk to you, meet with you, guide you in any of these senses. And we're also very happy to introduce you to people like Corey, you know, faculty at UCSF. So thank you for coming, and thank you, Corey, for being a great presenter and sharing with us so many wonderful things about the programs as well as your own research. And we look forward to having you again soon. With that, I'll stop the recording, and feel free to hang out if you have any other questions. Thank you.